Last Thursday, we gave a basic introduction to single-cell neuron scripting. We talked about a bunch of Python. It was a lot of material that was thrown at you last Tuesday. So we had some exercises that let you play with it, and we went through those exercises on Tuesday. If for some reason you weren't able to be there for that, I encourage you to look at the recording from Tuesday, because I think actually walking through some real world, semi-real world anyways, examples was very helpful. So before we get going today, does anybody have any outstanding questions or comments that they want to raise? All right, I'm not seeing anything. So in that case, let's just get started. So here's today's plan. So we're going to start out with some little discussion about some miscellaneous tools and techniques. We saw in Tuesday's exercises, as we went over them, we saw at the very end that neuron did not, so exercise 2, neuron does not by default, if you have Hodgkin-Huxley dynamics inserted in the channel, Hodgkin-Huxley didn't care about ion concentrations, and so therefore neither will neuron. However, sometimes you do very much care about that. If you're talking about ischemic stroke, if you're talking about calcium waves, there's lots of reasons you might care about intracellular, extracellular, ion, protein, cellular dynamics. Now, there's a whole ton of material that one could say here, and I'm not going to, but I just want to give you a little sense of a highlights and some things so you can look up some other videos that are available that go into that at some great depth. I'm going to give you a brief mention of Pandas, which is really useful for file storage. So you've done some simulations, you want to export some data to MATLAB or wherever to do some file manipulation. It's often very convenient to dump things to CSV or Excel or whatever, so you can open it directly there. Also going to talk about using Pandas for databases if you're doing parameter sweeps, especially if sort of parallel parameter sweeps. I personally had great luck using databases, and that way everybody accesses the same database, stores all the data in the same database, and then you go back later and visualize your results. Also going to mention briefly a library from my friends at the Blue Brain Project, EFO, which is the Electrophys feature extraction library. So if you want to sort of quantify some of the measurements, the spike trains that you're getting, that's a great library for that. And it's, by the way, has nothing to do with computational neuroscience per se. It's perfectly happy working with real recordings or neuron simulations, either one. After that, we'll spend a lot of time talking about shape and position. So this is the discussion on morphometric data. How do we load in a cell morphology? What does it mean? And then if time permits, Ted's going to talk a bit about events, networks, and integrate and fire cells. So that's sort of the plan for today. And like I said, so this first stuff I'm going to just go through really fast because I think you ought to know that it exists and its options. So Neuron supports simulating intra and extracellular chemical dynamics using the RxD submodule. So just like there's from Neuron import H, from Neuron import GUI, there's from Neuron import RxD. And so this allows you to define new regions. Oh, I should say, by the way, so all of these things I'm talking about briefly have a link that's associated with this. So if you go to this tiny URL, there is, I believe, a 2-hour-long discussion of what I'm going to spend the next couple of minutes just giving you an awareness of it exists. So this link is a YouTube video. You can watch it. It's me talking about chemical dynamics, how we model them mathematically, and how we model them within Neuron. So yeah, check it out if you're interested in that sort of, if you're learning more about that sort of thing. This is, I think, one of the great things that has come out of the last year and a half. We have a lot of recordings on a lot of topics. So even if we don't get to them in this course, that may be available for you to watch otherwise. So the idea with the reaction diffusion module, when we're talking about ion channels, we usually think about what happens just inside or just outside of the plasma membrane. But if we're talking about chemical dynamics, we may care very much about what's going on inside of the cell. And so the reaction diffusion allows us to define new regions. So we could define an endoplasmic reticulum or mitochondria. I could work in the far extended regions of outside of the cell where the cells live in 3D space and interact by, taking oxygen out of the extracellular space, right? So if you're talking about ischemia. So the RxD module provides a standardized way of doing this. With these 2 lines of code, I'm able to say, hey, look, I've got a cytosol somewhere. Well, we always have a cytosol, but it wouldn't have to be. And within that cytosol, we have calcium. And so if we do this and we use the RxD module, we're telling neuron that, by the way, we care about calcium concentrations. And since we care about calcium concentrations, I need you to monitor the calcium currents and update the calcium current concentrations based on the currents. And in order to do this, we need, of course, to know the ion charge. So there's 3 main parts to this, specifying any sort of chemical dynamics. And again, if you want to know details, please go back and watch that video that was linked earlier. It's a 2-hour-long discussion. But so just basically, whenever you're describing chemical dynamics, there are 3 questions you have to answer. First of all, where does everything occur? So we can define regions of our cells. So with RxD.region, and we can say things happen in the extracellular space. If we're talking about extracellular space, Neuron does not do like EM level simulation, but it will do sort of macro scale where you collapse all the details of the extracellular space down into volume constant and tortuosity. Again, all of this is explained in that other video. Who the actors are involved, what species, are there parameters, that sort of thing, and what reactions are specified. And importantly, you'll notice that all of these things are independent of if this is a deterministic simulation or stochastic simulation, 1D or 3D. Neuron's perfectly happy to use the same model in other contexts. Again, see the other video for details. But just as a couple of examples of the kinds of questions that you might want to be able to answer with this. So last time on Tuesday, we very briefly mentioned the circadian oscillator. So in the suprachiasmatic nucleus, proteins inhibit their own transcription. This leads to oscillations, as we see here, in the expression level, and then relatedly in the neuron firing pattern. But this happens over a time scale of roughly 24 hours. So this is a classic model. The link to the code is available here. It's a Google Colab thing. You can play with it. You can run it on your own and see how this goes. But this is the kind of problem. If you want to consider how does the protein interactions affect my electrophysiology, how does my electrophysiology feed into that? So this might be a good starting point of an example to use. It's also a fun one because this is one where, and this is why we talked about on Tuesday, where the units matter, where we're talking about things that are on a very different spatial and temporal scale than we're used to thinking about with action potentials. And that's fine. Zooming out the other way, we can look at the extracellular space. So if you imagine, so each one of these dots here that you see is an individual cell with Hodgkin-Huxley type channels. And we had a brief discussion about concentrations and Nernst potentials and all of that. But so what we're looking at here is extracellular potassium concentration. So here's my question. So this orange region is a region of elevated extracellular potassium concentration. If I have a Hodgkin-Huxley cell and the extracellular potassium concentration is higher, how does that affect my potassium Nernst potential? It's a question. Okay, let's do it this way. What is potassium's role in an action potential?
Helps in the repolarization phase.
Right. So its job is to bring down the membrane potential, you could say. And so it switches on when the membrane potential is high. And so how does it bring down the membrane potential? Which way does potassium go?
It's flowing into the extracellular space.
All right. So what if the extracellular? So that way and it can do that because there's more potassium on the inside than there is on the outside. So what happens if the amount on the outside is higher? How's that going to affect the flow?
Relatively less flow.
Relatively less flow, which means it's going to be harder for the cells to repolarize. And so we did this and we ran this simulation. So we just started out, this is not exactly Hodgkin-Huxley, but this model is available on ModelDB. I should have put the link. I'll look that up and add that to you. But we took an area of elevated potassium. It's going to make it harder for these cells to repolarize. So that means that they're more likely to be depolarized. But if the cells are depolarized, potassium channels are going to be open, and that's going to let potassium out into the extracellular space. So if I have too much potassium in the extracellular space, it's going to make my cells want to put more potassium in the extracellular space. And so we did this simulation, and like I said, the model is available on ModelDB. But what happens is that you see each of these individual cells. These are individual cells located randomly on a slice, but which is why it's sort of a mildly irregular shape. But you'll see that we get this region, expanding region of elevated extracellular potassium. So this comes up in, it's believed to come up in ischemia. This related to phenomena that come up in ischemia and migraines with spreading depolarization. So these are kinds of questions that you can answer, you can study using Neuron. And this is actually, don't even need network dynamics here. This is another form of cellular neuron to neuron interactions, right? It's just how do they modulate the extracellular space? So this is an example of why we might care about concentrations. And finally, I'll just mention very briefly that when we talk about the electrophysiology, we usually think of it as, a synapse is on a segment. We can actually be more specific with neuron, and we can say, if we're doing a 3D intracellular simulation, we can actually position it on an individual node, and there are examples for this in the documentation. I just want to direct you to that this is the kind of questions that you can ask, another type of question that you can ask. So how does it matter if two spines are near each other versus, in different orientations? How does it matter how the soma opens up? So Neuron's perfectly happy to let you address calcium and study, or whatever your favorite ion is, and study the effects of morphology on that. Which is going to bring us in a little bit to our morphology discussion.
Also, there's a tendency for certain types of ion channels to cluster together. So, for example, there are some calcium-gated potassium channels that tend to cluster next to calcium channels. And there's local interactions between calcium entering through the calcium channels and the calcium-gated potassium channels. And this is on the scale of, a few microns to many tens or hundreds of microns, depending on the sensitivity of the potassium channel to calcium. So there's a lot of important questions about neuronal function that's normal and disturbed neuronal function that can be addressed with exactly these kinds of representations that Robert's talking about.
Thank you. And so, like I said, there's that link at the very beginning of all of this, and you'll have the slides that'll take you to a 2-hour-long discussion of all of this if you want a full, fleshed out version. But I just want to point out some of these other issues and things that are within Neuron scope. I'm going to skip over this. You'll have this in the slides. It's an example of implementing. It's all you need, though. It's just a few lines of code to implement a model that incorporates some chemical dynamics. So here I'm using mass action kinetics. My little asterisk is this is probably not a very good model of calcium dynamics, calcium buffering, for various reasons that I can talk to you independently if you have questions. But at least it's a nice conceptual one. But this matters, for example, anytime that you're using a calcium indicator dye, it's working by calcium is binding to it. You can simulate those effects just like you can simulate the effects of whatever instrumentation that you're using to monitor in the experimental results using the linear circuit builder. So again, just want to point that out as a thing. So just to sort of wrap up our discussion on concentrations from the other day. Does anybody have any sort of I understand this was extremely brief, high-level view of all of that, but does anybody have any questions or comments on that as a concept?
Robert,
Yes, sir.
how does this interact with the extracellular mechanism that I'm used to in neuron when you're looking at current flows extracellularly? How do those two things interact?
So I'm going to punt to Ted on this. Do you have any? So that is primarily about yeah, I'm going to punt to Ted.
I could qualify that a little bit and give you a context instead of it being so wide open, but the interest really is in myelinated axons, and typically there was a double cable model that was done in neuron, I think in 2002, that's been used a number of times using the extracellular mechanism, but the small spaces between the axon and the myelin sheath are not modeled. So changes in the Nernst potentials and things like that are not modeled, but the behavior is electrically looks appropriate. So I'm just wondering if you could add this layer in as well.
Well, the answer is you would be having these are separate phenomena and they need separate mathematical representations. You don't automatically get ion accumulation in extracellular space with the so-called extracellular mechanism. That mechanism was a crude attempt to deal with the effects of extracellular non-negligible resistivity that might lead to local changes of electrical potential. If you need to represent ion accumulation in extracellular space, that's something additional you have to throw in, and RxD would be a way to do that. What this won't give you is electrodiffusion. Okay. I imagine that is possible to add to neuron in the future, but you could certainly represent restricted areas of restricted volume and restricted distribution of calcium or any other ion in intracellular or extracellular space, and have local accumulations that may equilibrate with other adjacent regions in ways that are representable either by ordinary differential equations or kinetic schemes. If you have a specific example in mind, we can talk about that separately. I think a guy by the name of Coggins did some modeling on that. I suspect that Terry Sinofsky might have been on one of those papers. The last time I saw Jay Coggins, he was on his way to Europe, where he was going to be doing something with EPFL, but it was at a meeting where he was indeed presenting a poster related to accumulations of ions in intracellularly and extracellularly in various normal and pathological situations involving myelin and myelinated axons or partially demyelinated axons. Are you thinking about those papers?
Yep. No, that's it. Thank you for that reminder, Ted.
Yeah. So you might look at Jay Coggins' stuff because he had some very interesting, complicated models that he was starting to work with, and I haven't followed that. I haven't followed those publications since, but I would doubt that he hasn't. I would doubt that he would have done something significant and not published. Okay, because he's a productive guy and works on interesting problems. Anyway.
So I do want to point out, so there's two sort of concepts of extracellular diffusion that we support. So one is this diffusion that's basically parallel to the cell membrane itself. And so you might imagine this is like a shell on the outside of the cell. And so this is one mechanism, but then, and the other option is, and so that follows directly along the path of the cell. And so there's no direct interaction between two neighboring cells. The other option is where they are living in this sort of 3D volume, wherein things can diffuse from one cell to another, or through various 3D space instead of right along the path. The path one probably makes the most sense in this like region of restricted diffusion that lies between the axon and the myelin sheath. I haven't really thought about it. Okay, so that was, any other questions about the chemical stuff and concentrations?
I have a quick question about the reaction diffusion module in general, and maybe this is covered in the video that you linked. But if I want to use it in a parallel simulation where I've got cells distributed across different nodes, can I use the RxD to have to simulate an extracellular concentration that can be seen by all of those different cells?
Yes.
Awesome.
I think if you look at, so Adam Newton, Newton et al. 2018, it was a methods paper, but if you look it up on ModelDB, there is a network model that uses MPI and extracellular diffusion.
Okay, great, thank you.
Absolutely. Any other questions? And just for the record, like intracellular MPI is a freebie because there's no interactions whatsoever, right, between what's happening on the inside of this cell and the inside of that cell. So that also just works, but the extracellular will work with the MPI mechanism. All right. One of the other things that came up in some of our discussions the other day was about, well, what do we do with the data that we generate? And so one of the things that I generally recommend is a library called Pandas. It's basically the sort of the go-to Python and data science library. It gives you an R, it turns Python into R, if you like R. So again, so here, this is a much shorter video, but this is about 30 minutes of me talking about Pandas at length. If you're interested in that, you can, more systematic presentation of what all you can do with Pandas. Here's a link, and again, you'll have these slides. But fundamentally, the two most basic things that I imagine that you'd want to do anytime that you generate data is to save the data to a file and load the data from a file. So basically, if you have T and V or vectors that you've recorded, you can generate what's known as a Pandas data frame in this format. So it's just the header, and then the header colon the variable, the next header colon the vector. And then just that once you have a data frame, you can dump it to CSV, just give it the file name, and then it'll generate a CSV file, which is a comma separated variables file, and that you can open up in Excel or what have you. MATLAB, obviously, and pretty much everything's happy loading CSV files. It's literally just the numbers separated by commas. You can also read in data with CSV from Pandas. So we talked about, like, maybe I've given some sort of complicated stimulus pattern and I want to feed that into a cell using a vector play. Well, then I can load in data from a CSV file using pandas.read CSV and then just the file. And then I can grab an individual column and turn it into a vector if I want by h.vector parentheses, my variable name, and then square brackets to select the column that I'm interested in. So this would give me back a regular neuron vector that is just the T column or just the V column or whatever data I want to feed in to my model. And I should say, so Pandas is extremely flexible and extremely consistent. So just like there's a to CSV method, there's a to Excel method, there's a to JSON method, there's a to HDF method, there's a read, you know, all of these things. There's a read HDF, read JSON, read Excel, read SQL. These are all things that you can do. So you can work with pretty much any data type interchangeably using Pandas, and you can use this to bring data in for your simulations or export them to other tools for analysis. Or even just save, here's another thing, let me just point this out, and just save them sometimes for yourself, right? You don't necessarily, you know, if you're running a whole bunch of simulations, so for example, you'll remember that we had the parameter sweep exercise in the homework from last Thursday. And so we did the same thing over and over again, except we just clamped voltage at different values and we just used a for loop. Well, maybe I want to save all of this stuff as you know, these were very quick and ran in seconds, but maybe I want to save all of these things. Maybe it's a much more complicated model and I want to save all of these things as I go so that I don't lose data in case my simulation fails or something like that. So again, so even if you're working solely in the neuron environment, sometimes it's useful to be able to save data, bring back in data. Just keeping this out. And Python, by the way, if you don't know, has a built-in multi-processing module, which you can use to parallelize parameter sweeps in just a few lines of code so that you split it off so that instead of having neuron do one thing at a time, you know, if you've got an 8-core machine, neuron does 8 things at a time.
You have 8 different instances of neuron, do one thing at a time.
Yes. And it's all controlled by one single thing. So you can, it's effectively a for loop. It's expressed in a way that's effectively a for loop, but now all of a sudden, everyone runs independently of each other. And so they each calculate whatever simulation and they generate some data that you can then go back to do this.
So suppose you have a set of, say, a couple of hundred different simulations you want to run, and you've got 4 processors. Can you just throw that 100 different jobs up, and your 4 processors will just work away happily, picking up things and chewing them and spitting out results, and then picking up a new thing, et cetera, et cetera, et cetera?
Yeah, so this is basically what's known as a worker thread type model, except with processes. So I can put up an example of this in the Slack later of exactly how you would do that, but yes. All right, and so let's say that you are doing this. So you generate all of this data. What do you want to do with it when you're done? And so, you know, because if everybody's trying to write to the same file, what if they're all trying to hit the same file at the same time? Or, you know, it comes out in a weird order, or there's any of a number of ways that you could imagine that your output could be a little bit garbled if you've parallelized things. And so my suggestion, you're free to do whatever you like, it's Python, but my suggestion, Python comes with a library, Python comes with a built-in databases module, SQLite 3. And so what you can do with Pandas is you just define your columns. So here, this is a slightly modified version of an actual multi-dimensional parameter sweep I ran where I was experimenting with dx and length and diameter and a parameter. And so I had a whole bunch of things that my model might depend upon. And I calculated some values. So like, let's say I calculated a spike half width, like calculated some other values. Personally, I also like to store the runtime. It really doesn't take anything. So perf counter gives me a high resolution reference to how long it's been since Python was launched. And so I can use that to subtracting when I started from now and get a decent estimate as to how long an individual simulation ran took. It's good to know how long one of these things takes so you can estimate how long the whole thing's going to take. But so in any case, so I do, you know, all of this is happening inside of a for loop. I'm generating my data. And then whenever I go to write it out, these 3 lines is really 2 lines of code here. Whenever I've done my calculation, whenever I've done my simulation, I'm going to connect to my database. And the cool thing about SQLite is that the database is actually just a file on your system. But it's a file that it protects in magical ways so that there's no collisions that, and everything just works if multiple processes try to access it. But so in any case, so with SQLite.connect and then my file name as con, so it's just going to give me an object called con, it's my database connection. And then with pandas, I have my data frame, data.toSQL, I give it a table name, so data, my connection, and then I'm just going to tell it I'm just going to append onto that table. And this is rather formulaic code. You just identify your columns, you dump it out to, you use this to store, to append onto the table. And then so you're generating tons and tons of simulations, tons and tons of stuff. You could imagine maybe you're running this on a cluster, you're able to generate with MPI if you want, you're able to generate a lot of data. And then the cool thing is, if you do it this way, you can logically separate, you know, maybe this takes a long time to run. Maybe it takes a while. Well, if it's going to take a while, you can do 2 things. First of all, you can make your program such that it checks the database and says, hey, have I already run this one? And if so, skip it, right? So that way you can just stop if your program dies. Like there's always a timeout when you're doing these HPC systems. So you could just resume. And so just check the data, see if you've already run the simulation, check the database, see if you've already run the simulation. You can also visualize stuff as it comes in, right? Just go ahead. It's all writing to a database. You want to read from a database, go ahead, read from a database, and here's some code to do that. And you can do just sort of give me everything that's in the database. This is an actual SQL command if you know that. If you don't, it doesn't matter. This just gives you all of the columns. Or you could do some really complicated things with databases like you do to say the kinds of data that you're interested in. Maybe I'm only interested in certain columns, or I'm only interested in data where I got some results in a certain range. And, you know, SQL's perfectly happy doing that kind of a restriction for you. If you know that, great, you can use it in this format with neuron data. If you don't, just grab all of the columns and do everything in Python. But the point is, I'm sorry, I got a little distracted, I was too excited. The point is, if you're running a long running simulation that's calculating a whole lot of values, right? Because maybe you're doing a parameter sweep, you know, how does this model depend on the sodium channel? Because you know what? You don't actually know that sodium concentration, sodium channel density that well. So it's probably a good idea to see the sensitivity of your models to any individual parameter, right? So you're gonna wanna do these parameter sweeps just to make sure that you're not in some like weird special niche domain. But so if it takes a while to do this, what you can do is just go ahead and have a separate script whose job it is to visualize things. Go ahead, pull from the database, whatever data exists, plot the data that you have, you know, so there's no point, you don't want to be in a situation where you have to wait for your entire simulations to finish before you know if any of them worked. So that's another advantage of sort of dumping having one script that does all the simulation, dumps to a database, and another one that loads from the database to make a pretty picture. right? You can check it as it goes instead of waiting for the end. So just a suggestion on working with data. And that's true if you're doing a networks model or just a single cell. Does anybody have any, like I said, and there's a whole systematic description of pandas, but I just wanted to point that out as a life tip. Does anybody have any questions or comments on any of that? Okay, one last thing before I want to get to the main, before we get to the main feature of today's presentation, one other thing that I want to give a shout out for is the EFEL Electrophys Features Extraction Library. This is from my friends at the Blue Brain Project. And so what this is so you've generated a spike train, right? But how do you actually, what can you measure out of a spike train? And how do you actually, you know, build formula that calculate a whole bunch, you know, I think these half width of a spike came up at one point. Well, how do you actually code that? And so EFEL basically does all of this for you. And so you can install it. It's not included with Neuron, but it's a single line pip install EFEL. Documentation link is there. And if you do one of the, and so you load this library by, you load this library with import EFEL. And then if you want to see what all it can calculate, you just say efel.getFeatureNames. And so it's going to give you a whole long list, and I've cut this off at the letter C. It's going to give you a whole long list, so the whole thing's maybe twice as long as this, of features that it can calculate, you know, like the action potential half width in here somewhere. You know, features that it can calculate based on a spike train. And if you go to the documentation site and you look up every single one of these has the formula involved available in the documentation, so you can see exactly how it interprets it. So this may spare you if you want to sort of compare qualitative results from the literature. You know, you don't care so much about the spike. The actual details of the spike train may not be that interesting. If something, you know, spikes at time 3 versus 3.05, who cares? I mean, it might make a large second, mean square error, but if you're actually looking at, well, what if the spike widths are right and the amplitude's right and whatever else you think is important about the rise time is right, you know, maybe that's a pretty good model, even if the mean squared error is not so good. So in any case, EFEL does a lot of this stuff for you. But what is the first rule of using somebody else's code? Because this is using somebody else's code.
Trust but verify.
I like it. Trust but verify. So what do I mean by yeah, so what do you mean by that?
Make sure it does what it says it's going to do. Don't just take it at the programmer's word blindly. Verify it.
Absolutely. And the other thing that I would suggest by this is don't just take your interpretation of what the program, like one of those things you ever find that, oh yeah, it does exactly what they said it did, but that wasn't what I heard, right? So it's always very important to go back and make sure that things work the way that you think they do. And so here's what I would do, for example, if I was doing this. So here's a simple code. I coded up a thing. And I think we know all of this stuff at this point. We load Neuron, we load our units, we load our run control library. We construct a model. So it's going to have a section, a single point model, 10 microns in length, 10 microns in diameter. It's going to have Hodgkin-Huxley channels. It's going to have an exponentially decaying synapse with a certain reversal potential. Here, it's a very fast time constant. Why not? we can, so here's a model of our cell. This is a model organism. And to this model organism, we're going to say that we're going to have a set of stimuli. They're going to come in starting around 5 milliseconds. I'll get 10 of them, I'll get 10 of those in total. And they're going to average 10 milliseconds apart, but I'm going to use a noisy spike train. So what that means, if you set noise to be true, this is generating a Poisson process where the mean time between spikes is 10 milliseconds. And for various reasons involving math and statistics, there are reasons to think a Poisson process is a reasonable model for when cells get it. We build a netcon to connect a stimuli to the cell, to the synapse itself. We give it a certain synaptic weight. We talked about the weights. We actually looked at the implementation of the exponentially decaying synapse in a previous discussion. We record time, we record membrane potential, and we say go. And that's it. That's our model. So I'm generating a slightly interesting set of spike trains because it's going to be driven by this random event. And here's the full, well, this is the full code, but if you want to run this on Google Colab, here's a link that lets you play around with this interactively. And so this was my spike train that I got out of this. And you'll notice, by the way, so it fired, and then I got this funny little dude where it didn't fire. What happened there? What happened with, why did I get that failure to fire?
Might have hit it during the refractory period.
Yeah, that's my guess as well. I mean, I didn't do anything to prove that, but like, yeah, I'm going to say probably. And I think it looks like there's actually a second bump here. I don't know if you can see it on your screen, but so it might have hit it twice during the refractory period. It wasn't quite ready for it. And so it failed, but then later on, it's able to get another stimuli and fire an action potential. So if I want to quantify this, so here's some example code. These are the 4 pieces of data that the Electrophys library here needs. It needs to know the time values, duh. It needs to know the membrane potential, duh. It also needs to know when it should be looking. So we'll just say that I want you to look between time 5 milliseconds and 100 milliseconds. So what you do, so this is one particular spike trace. You can actually give it a list of such objects, and it'll give you a list of results in response. So like if you have multiple cells, right, you might ask it to do the analysis for all the cells in one go. And then you just say efel.get feature values, and then your trace data, the time, the potentials, and when it should be looking. And then whatever you want to know about from that long, long list of things it can tell you about. So I say, well, what's the action potential amplitude? What are the interspike intervals? What's the spike half width? What's the action potential begin time? All right, action potential amplitude. Well, there's a number of, there are 5 spikes, so I get 1, 2, 3, 4, 5 results. That's a good sign. 103, yeah, sure, fine. It went from a little below -60 to about 40. Yeah, I'll believe that was a height of 103. And so there's some variation between the individual spikes depending on the state of the cell before they fired, but it's pretty consistent. We can get a list of the interspike intervals: 12.7 milliseconds, 17.5 milliseconds, 25.5, 15.2. Yeah, I can believe that looking at the picture. I wouldn't believe it if I'd need to see the picture in order to decide if I believe it. Half width, sure, we can figure out if we go up about halfway. How wide is it? Sure, why not? But here's one little gotcha. What do you think about the action potential begin times? I personally, when I first ran this simulation, was a little surprised.
The middle one seems off. It looks like it's picking up the failed stimulus rather than when it actually spiked.
Exactly, right? So something's happening there. It never goes back down to rest. So it's lumping this entire third thing sort of together. And maybe that's what you want. I don't know why, but maybe it's not. But it's important to be able to say, to have, because we had this test, we were able to say, well, is this what I want it to mean? Like, they can give me a formula, but if I don't look at a picture where weird things happen, I don't know if even just looking at the formula, is it doing what I think it does? So, I'm sure it honors its formula, but it may not understand our intuition as to what that should mean. So I think this is a reason to do the verify, run some tests, make sure that your tests have something weird happen, and see how this handles the weirdness. And this has nothing to do with them, right? Like I'm recommending this library here, but this is just in general when you're using somebody else's code, test it before you use it in your own code with something simple.
I would submit if you showed this figure to 10 experimentalists, not a one of them would agree with that third begin time. Every last one of them would say it's more like 40 milliseconds. So I think this is a failure of the algorithm. It might fail with this particular model. It may not fail with other models. For example, this is a squid axon spike, remember. If they were testing this entirely on vertebrate neuron type channels producing vertebrate neuron type spikes, that peculiar hump, that third response, may not occur in a model of most of the vertebrate type action potentials. So for that kind of data, it might work just fine. And this, of course, is the nature of that project. I mean, they're basically focusing on vertebrate systems. More to the point, mammalian systems.
Yeah, so especially that's another good point. Like if you're finding code, and this is true for ModelDB as well. If you find code on ModelDB, what are they using it for? And does it apply to your system? I mean, if they're studying Alzheimer's, well, maybe there's something special about the cell that makes it in that domain. It may not be appropriate for a healthy cell model. So if you're using tools that are designed for one environment, you've got to make decisions and good calls. And again, test before you use, because otherwise you may get results that don't mean what you want them to mean. And I just want to point out, so related to that, so you probably noticed as you've dug around through the menus that Neuron includes a multiple run fitter to try to optimize things. Of course, one of the great things about using Python with Neuron is you can use pretty much any machine, any optimization library out there has a Python interface you can use. So also from the Blue Brain project is this tool called BluePyOpt, which you can use to optimize parameters. And it's especially designed to work with Neuron, and it's especially designed to look at those kinds of features that EFEL is good for identifying, like spike half width and rise time and that sort of thing. So I just want to point out that exists when you're trying to fit data, and you may want to think about what's the actual data that you want to fit, because it's probably not the literal traces, but rather some property thereof. All right, so any questions on that before we get into neuron morphologies themselves, which we've hinted about? All right, let's go on and talk about neuron morphology. So most of the stuff that we've talked about, here's a single section model, or here's a model with several sections that are connected together. And so obviously when you do that, you do get to explore, for example, the difference between synapses that are far versus near. Some phenomena are just fundamentally local. You may have cells that don't generate action potentials, or neurons that have dendritic synapses, which is a phenomenon that occurs in certain cells in the olfactory bulb. But there's a lot more that's going on, and the morphology plays a very complicated role. And here's just a few papers that I came across when I was digging this through. Right, spines and dendrites are known to play big roles that directly affect dendritic computation by modulating how inputs combine locally. Morphology matters is the general claim, at least depending on certain circumstances. We've already seen from the graphical tools that you can go to graph shape plot on a neuron and pop up an interviews window that allows you to manipulate the shape, view it in 3D, rotate it, etc. So you can also do this programmatically. So if you have a cell here, here my cell, remember we said it was a good idea to make our cells classes, because if we make our cells classes, we can make as many copies of these as we want. And all we have to do is use 2 extra lines. So yeah, so I defined a cell, it's got a main and 2 dendrite sections, they're all connected together, a certain diameter, a certain length, and I can just go ahead, instantiate my cell. If I do PS is h.plot shape, and I pass in true, it'll pop up an interviews window. So the same exact interface that you get if you do it from the command line, from the same exact interface that you get if you do it from the GUI. If instead you pass in false, you can then manually construct it. And so I can do ps.plot on plotly, and I'll create a plotly window with plotly graphics, and then you can show it. You can also highlight certain sections on this. So if I have a point process in various locations, I might want to mark those off in my plot. In addition to plotly, you can do matplotlib, and because these are 3D graphics, our options are somewhat limited, and I think those are the only 2 that we currently support besides interviews. By the way, also I should point out, this question came up really early on in the Slack. If you've created a plot shape and you've positioned it and you've done whatever you want to it, and so that plot shape, let's say that plot shape object is called PS. Then you can do ps.print file, and then just give it the file name, and it'll generate an encapsulated PostScript file, vector-based graphics representation of whatever's in your plot shape window. And yeah, one of the sort of unfortunate things that I will admit, because you may have noticed in the graphical interface, this is called a shape plot. On the programming side, it's called a plot shape. I don't know why. It just is. But once you have that, regardless of what you're using, if you're using interviews, matplotlib, plotly, doesn't matter. If you have different voltages, let's say, you can use whatever your plot shape object is, .variable, to select the thing that you want to plot. So here I'm plotting voltage, and I'm scaling it between -80 and 80. And so this is going to give me sort of a heat map distribution of voltage, where I've set it as I did before. I can show it, obviously. You can also pass in, like if you defined, intracellular calcium in the endoplasmic reticulum using the RxD infrastructure, you can pass in that RxD object, and it'll plot intracellular calcium in the ER in your plot shape. Cool. So one thing that you may have noticed here, this is a bit of an undetermined problem here. It's decided that this is what the cell looks like. Does it actually know that? Is this what the cell looks like? Like, does everybody see the issue?
Well, you haven't specified any shape. It doesn't have to look like anything. It doesn't have to look like any representation like this. It could look quite like, you know.
Right. The only thing that neuron knows at this point, because that's all that we've told it, all we've told neuron at this point, yikes. All we've told neuron at this point is that there are 3 sections. Neuron does have defaults for the links, so we know the links, we know the diameters, and we know who's connected to who. But what that still leaves us is a huge potential for the angle and the orientation and the position. So there's a ton and ton of degrees of freedom that we've specified and that we've not specified. Also, when we're doing this, if I'm setting a diameter, I'm saying that everything's a cylinder. No one doesn't care. Cells certainly aren't cylinders. So what we can do instead, and we saw this last Thursday briefly, we can specify our own 3D points. So this is an example adopted from the neuron documentation here. This is mildly absurd. But what we've got is we create a section. We make sure that there are no 3D points already there by point 3D clear. And then I can define whatever my rule is. And point 3D add on my section is just going to add a new, I'm going to say, hey, I'm going to go through this XYZ diameter point, because the diameter is always going to, you know, those are the 4 variables that are defining the shape of our tree, that is the neuron, right? So an X coordinate of 200 sine X, a Y coordinate of 200 cosine X. I'm going to put Z as 0 just because, and a diameter of 100 times the sine of 4x. And then now, if I create a shape plot, which I can actually also do with shape, but it's slightly less capable than the plot shape, you know, and I pop that up, this is what I get. So it can be arbitrarily complicated. It has to be radially symmetric, but other than that, the diameter can vary continuously. And you'll notice that this, by definition, only has 1 segment, but the diameter is constantly changing throughout the segment. So be it. By the way, there's one sort of well, there's at least one fundamental thing that should concern you about this geometry for my cell. What happens when the diameter goes to 0? Or in general, what happens if the diameter is very small in a neuron? What are the implications of that at some point? Is it easy to flow through things where the diameter is small? Well, it's a leading question, right? If the diameter is small, it's hard, there's less paths for current to flow, which means it's sort of a pinch point in currents. And so it's actually one of the things that I'm going to suggest in a little bit when we look at loading in morphologies that we found somewhere else. So make sure we don't have any places where the diameter gets really, really small, because those are going to block current flow. And those are probably errors if we come across that in a morphology reconstruction. So I just want to point this out. And there's a whole bunch of other methods that you can look at. And if you look up point 3D add, you'll see the whole list of them in the documentation. So fundamentally, our shapes, so now what all defines the shape of a neuron? We've got a whole bunch of X's, Y's, Z's, diameters, and we need to know who's connected to who. Is there anything else that we need for a shape that's appropriate for the electrophysiology? Well, that's really it. And so one of the things, so then over time, starting in, I don't know, certainly by the 80s, people were able to start recording this information from microscopy images. And so one format that people use, it's widely used, is what's known as an SWC file. And it's just one of several morphology formats that Neuron supports. And so basically what's happened is optical imaging, and somebody's gone through, could be somebody or some algorithm, has gone through and tried to track a neuron through a whole bunch of Z slices and said, well, okay, it is here and it's this big when it's here. And then that point is connected to this other point, where it's here and it's this big. And so by the time somebody or some algorithm has done that, you end up with data that looks more or less like this. So here, these formats have an identifier. These are just going increasing. A type. So this 1 indicates the soma. Type 3 indicates dendrite. Doesn't really matter. But an X, a Y, a Z, a diameter, and then information about what point this point is connected to. So the first one is the soma. It's not connected to anybody, but our first point of the dendrite is going to be attached to the soma. Our second point of the dendrite is attached to the first point of the dendrite, and so forth and so on. And so this is one common file format for representing morphologies. And so if you can get your hands on, you can either generate one of these or otherwise get your hands on this. You could use all of those 0.3D infrastructure. You could read these files. You could use all of the 0.3D infrastructure to construct the sections. And then there you go. You've got a model inside Neuron. That sounds like a lot of bother though. So let's make a GUI for it. And so it was. So there's a graphical interface, and I'll demo this in a bit. But there's a graphical interface that's available under Tools, Miscellaneous Import 3D, that allows us to read in a variety of file formats, including SWC. It'll let you know if it sees any errors, certain types of errors, namely things that are disconnected, but not necessarily pinch points. And I'll mention some other errors, common errors. But it'll detect at least disconnected things, and it'll let you know about them and give you the ability to fix them. But so you get this graphical interface like this, and then under export, you've got 2 options. One is to export to a cell builder. So then you get a cell builder that you can then use to insert channels and do all of your other graphical stuff. And this saves to a session exactly the way you'd expect it to. One minor caution for you: when it's inside the cell builder, it will appear to have a simplified shape. It does not. The actual data is still there. But Cell Builder for this, because some of these things were built a while ago, Cell Builder uses a simplified representation of the shape just to make it faster. But so you can export this to Cell Builder or export it directly to a cell, top level, and then you can work with it there. And then of course, if you've done this, you've exported to a Cell Builder and then instantiated from there. You could save it as a session file and then work with that. There's a whole, I'll work through an example of this, of using the Import 3D tool, but there's also a tutorial available that you can check out on the Neuron website. And I'm going to generally suggest that any time that you find a new morphology, you're probably going to want to start out, as Mandy said, trust but verify, right? So here's something new that you got from somebody else. Is it appropriate for your purpose? It may be very good for what they wanted to do with it, but it may not be appropriate for your purpose. So if you start out, don't dump it in, if you've gotten a new morphology, don't just start out building a 10,000 neuron model with extracellular diffusion and the networks and gap junctions, and look at the cell, look at the individual cell, because just because you get the cells right doesn't mean that the networks are going to be right. But certainly if the cells aren't good, the network's not going to fix it, right? So look at the individual cells, make sure that they make sense. And it's really easy to toy around with things with the graphical interface. And I invite you to do this as an exercise, and we'll have formal specification of the exercises in a bit. Does that make sense? You know, look at these things, see if they make sense. I'll give you some pointers of what to look for in a second. But I do want to mention, since this is primarily about programming, while I suggest you start out playing around with the cells graphically, you certainly don't need to use the graphical interface to load stuff in. Here is an example of a cell class wherein we've defined a morphology, a function that loads a morphology, and it's a very stylized representation. These 4 lines are basically what you need. You're always going to use an import 3D SWC read. Well, that's a slight lie. That's for if you're using an SWC file. If you're using a Neurolucida file, there's an import 3D underbar Neurolucida 3. If you're using eutectic, there's an import 3D underbar eutectic, and so forth. And this is one of those great things about dir, right? You can just type, you know, if you've loaded the import 3D library, h.load file import 3d.hope, you can just do a dir of h, and just see all of the things that start out with import 3D, and that'll tell you, well, that'll give you your list basically. So you're always going to construct a reader. You're going to feed it whatever file name that you want to read the morphology from, and then once you've done that, you invoke that, you ask the GUI to do a thing. So you pass in whatever you've read. You send in false, which is a flag that says, I don't actually want a graphical interface, but I want the tool to do a thing. And here my tool is going to instantiate the cell, instantiate the morphology that it's loaded inside of me, inside of self. And what that means is, once that's happened, you know, so here I've got an SWC file, C91662. I run this code. I have defined a type of cell that is pyramidal, doesn't actually create anything until I instantiate it with PYR as pyramidal, open close parentheses. Once I've done this, PYR has a bunch of lists of sections. It's got a PYR.apic. Those are the apical sections. It's got a PYR.axon. That's the axon. It's got a PYR.soma. That's a list of sections that are associated with the soma. It's got a PYR.all. Well, that's a list of everything that's involved in that one particular cell. And every section knows its name and knows its cell, and it does the right thing. But so this is how we would do the same thing programmatically. Like I said, you can basically just copy paste this code into your own model. So we can load it in graphically. We can load it in programmatically. We've said that there are various things that we should test for, but we can't test it until we actually have one. So how do we get our hands on one of these suckers? How do we get our hands on a reconstructed morphology? Because that sounds like an awful lot of bother to take a whole bunch of stacked images and finds X's, Y's, Z's, and diameters. Anybody know where we can get our hands on these? So one option is to have a friend who likes doing this, right? Another option, though, is the website neuromorpho.org. So this is actually a screenshot from a year ago, so I assume there's somewhat more than this. But you'll notice that they have 131, a year ago, they had 131,000, almost 132,000 neuron reconstructions. All right, representing well over 500 different cell types. They have from many species. So it's an incredibly valuable resource. So you can go in there, you can specify exactly the type of thing you're looking for. There's so many of them that even if you're looking for a specific file format, that's probably a fair thing to do. And by the way, some of the file formats have different features that the other ones don't. So if you're using SWC, the somas are going to look like tubes, because that's how everything is, right? It's a point in diameter, a point in diameter, and then it's just going to have the cone that connects them. If, on the other hand, you're using a neurolucidar file, one of the things that you'll see in neurolucidar files is that people tend to trace the soma outline. And so if you're doing anything that depends on the shape of the soma, then that might be a thing that you'd want to use. Maybe you want something that has a neurolucidar file that has the soma outline. Now, if I'm talking, just as a side on that, if I'm talking about the shape of the soma mattering, am I thinking about electrical signaling or chemical signaling, and why? If it's obvious, you can say, that's fine too.
You're talking about chemical signaling.
Yeah, why isn't it for electrical?
Because we represent it with the cable equation, and it's very simplified.
Well, yeah, and the soma's big, right? So like, there's not a whole lot of resistance inside the soma. But chemically, if the soma is big, well, then that's a lot of space that we have to move across. So, yeah, so it doesn't matter if you're looking at electrical signaling, doesn't matter if you're looking at chemical signaling. But so anyway, so once you find an example, what does it look like? So here is the top of a neuromorpho figure. So there's a ton of metadata that lies below this that will tell you all kinds of things about path lengths and angles and lots of other things, what features are present. So this is one that respects the diameter. It's supposed to have meaningful diameters, meaningful third dimension, and meaningful angles. And the reason that this is important to look at, by the way, is because you know, this is a resource of neuron morphologies and it's whatever people have studied, and maybe their research didn't require looking at the third dimension, or maybe their research didn't require knowing the diameters. But if we do know the diameters, you know, if we're doing if we're doing a simulation, we probably very much care about the diameter. So you've got to look at everything to decide if it has the features that you want, if it's good enough, but at least they do provide some information. Here we've got all the dendrites, but the axon is incomplete. That's probably fine, right? Because the axon, as long as you're looking at a healthy cell, the axon should propagate signals relatively faithfully. So maybe I don't need to know. It doesn't do me a whole lot of good to actually explicitly simulate the entire length of the axon. In any case, so they have, every single entry has whatever the original data format is. So it could come in as SWC, it could come in as neurolucidar, it could come in as any of a wide variety of original data formats. And what Neuromorpho has done is they've standardized every single entry, all 131,000 as of a year ago, have been standardized and converted into SWC. And that's why I made SWC my example for everything, just because it's always available if you're getting it from neurolucidar. So you can go on there, you do your search, you download it, and you're good to go. So maybe this is a nice moment to, let's just do one real quick and bring it into Neuron so that you see the process of this. So in order to do this, we need to find, oh, come on. Maybe we'll do one real quick if I can get a web browser up. What is happening? So if we go to Neuromorpho, I said, okay. So if we, this has nothing to do with Neuron. This is entirely my computer being slow. We go to neuromorpho.org. You know, so where are we at? We're almost 150,000 neurons now. But I could, let's just look at by animal species, just real quick here, just to see the kinds of cell types that we might have. So they've got, you know, 46,000 reconstructions from a mouse, 42,000 reconstructions from a rat, 33,000 reconstructions from a fruit fly, almost 3,000 from a monkey even, right? So what are our, well, that may not have been a good idea. But in any case, so we can take this, we can pull up whatever we want. So here we just found this particular one, kind of from randomly clicking around. But this came from our monkey, this is a macaque. That was somewhere between ages, this is a male macaque, somewhere between ages 7 and 12. It was originally represented using Neurolucida. I have a whole bunch of metadata about the soma, the branch angle. And so forth. We can see what does this one say is available. Wait, where is it? It's got meaningful diameter, meaningful 3-dimension, and meaningful angles. So it sounds certainly promising. If I want to look at it interactively, I can click on pull up the WebGL viewer here. And what did I do? And click on the WebGL viewer. I can spin it around. It's awfully flat if you ask me, but I can look at it from a variety of angles. I can zoom in, I can see how things are. You do see that the SWC, it has standardized everything into basically tube-shaped somas. Again, this probably doesn't matter if you're looking at electrical signaling, but it does matter if you're including chemical dynamics. So you say, I like that one enough. Let's go ahead and download this guy. So I'll just take the standardized morphology file. It'll give me this SWC file. Again, it's X, Y, Z's, diameters, and parents. I can just save this on my computer somewhere. Yeah, so I'll just throw this on my desktop for simplicity. Actually, I'll throw it in my downloads folder. All right, cool. It's in my downloads folder. So now if I were to open up a terminal and I would open up a terminal, go into my downloads folder. Again, you already know how to do the actual launch. All I'm going to do is launch Neuron. So your individual process may vary depending on if you're on a Mac or Windows or whatever. But in any case, I will just do Python from Neuron import GUI. And then it's on my other screen. Hold on. So here we go. I get the Neuron graphical interface and go Tools, Miscellaneous, Import 3D. I'll click Choose a File. So here's my Import 3D tool, choose a file. And somewhere in here, there's this SWC file that I just loaded. And boom, here it is. This is the morphology that we got from Neuromorpho. Does it look right? Well, let's go back to our pretty picture. Here's what we saw from Neuromorpho. Here's the shape that we see in Neuron. I'll turn off the show points, and there you go. So it's definitely the same cell. We pulled up our cell, and if I wanted, I can either transfer this over into the cell builder or instantiate it directly. And then the reason for doing this is maybe I want to do some tests to decide if this is right for me. Inside the cell builder, just to show you, you'll see that it looks a little rough. It's preserving the overall shape of the cell, but it has simplified all of the sections down into sticks. That's okay. Don't panic, because if I hit that continuous create and then I look at my shape plot, I still have all of the details of the original model. It's just that in my cell builder, the shapes are simplified a little bit. That's all. So then I can use this. I can save a session file. I can insert channels to do analysis. I can do all of those things. So it's the basic process of at least bringing something into Neuron. Makes sense. All right, so if so, let's move on. So just to look at this, yeah, question? Or Ted, comment, or?
Well, just that there's a couple of detailed tutorials about using the import 3D tool on Neuron website under the documentation page. There's a link to that. So if you do have questions about it, you can find a lot of details there.
Yeah, and the link is included as well on this on the slides. So if you have it, yeah, when you have the slides, you'll have it. But again, so fundamentally, do remember that not everything has been made for you. So various things may, it's not necessarily intended to be used as a simulation. And so it's very important to go back to figure out why this model exists, right? Is it, and what was done? There's metadata in Neuromorpho. It's also a good idea, though, if you're going to use something, read the paper, see if they say anything, any special conditions that are going on that apply to this. Maybe, if they were studying a disease and you're not studying that disease, you probably don't want that model. You're going to want to check, various factors may have happened in the process, how the cell was stained. Different cells are going to fill differently. Well, sorry, different stains are going to fill a cell differently, and that's going to have implications on how things are visualized. You may have these models. It happens where the diameter is reported to be very small. So that's the thing to look for. So if I was just to suggest, start out by looking at whatever Neuromorpho's metadata says. But again, trust but verify, always do your own tests, look for disconnected or barely connected sections. So one way of doing this is inserting a passive conductance, lower the actual resistivity and leak conductance. You do want a little bit of leak conductance, because otherwise the cell will charge no matter how small it gets. But as long as there's a little bit of leak conductance, if you inject a large depolarizing current, if there's a pinch point where it goes down to nothing, the current will just flow out the leak instead of lighting up the entire cell and having membrane potential raise everywhere. So if there's no pinch points and you have leak currents and a large depolarizing, you'd expect to see the membrane potential go up across the entire cell. One other common thing to look out for is, you'll notice that when I was on Neuromorpho, I went, and one of the first things I did was I popped up that WebGL viewer, and I said, well, how does this cell look from different angles? Obviously, you can do this with a plot shape as well. Is there any sort of z-axis drift? Is there a systematic bias where the cell's just kind of migrating off that way? Or sometimes you'll find cells where there's an abrupt jump. Looked for that. Obviously, also, again, neuromorpho does report about diameters, but you should still check them to see if you think they're reasonable. So make sure that the diameters are non-constant. Are they spanning reasonable values for the cell types that you're looking at? So there's lots and lots of things that you might want to test before you do this. And all of these things can be done either programmatically or with the graphical interface. And I'm going to encourage you to do that for the homework. I'm just going to point out some references. If you are interested in all of the things that can be problematic in data quality of neuron reconstructions, here's a couple of papers that you may want to check out. They're a little older now, but their points still stand. There's also a relatively recent tool called MorphoUnit that's for morphology testing as well. You may want to check that out. All right, so that's the actual act of bringing in a morphology. Any questions on the act of bringing in the morphology? And quality issues with that. So sort of the next thing we were going to want to talk about is networks and multiple cells, but I just want to talk about, before we even connect cells and think about how synapses work between cells. So let's just imagine what, yes, sir. Question.
I have a quick question. First of all, thank you so much for a very informative lecture. The question is that if I want to say that use a cell from human hippocampus, not the monkey's hippocampus, is there any possibility that I can find something in the resources that you already introduced us? Like, I like to see that, is there any human hippocampus morphology and also the monkey's hippocampus? And can I see the differences between these two? Am I expecting to see the differences?
So there's actually some papers that have looked at some of the morphological, but also ion channel expression differences between these cell types. So I'll point out, I think Ninad Seston at Yale has been involved in some comparisons of humans versus monkeys in terms of the channel differences between the between cell types. But just looking at this here, looking at neuromorpho, I see that there's 7,000 human models. So I assume, and let's find out, we can go in and do a search on the metadata. I'm looking for humans, found them. What do we want? Brain region, hippocampus, CA1, I guess. I'll grab principal cells. Almost there. Come on, looking for pyramidal. Almost PM, projection, pyramidal. There we go. So this should be human, hippocampus, CA1, pyramidal cells. Let's see what we've got. So according to this, we have 54 human CA1 pyramidal cells. We can pop them up, which shows summary here. And it gives us, conveniently, it gives us a nice, convenient, and actually human readable link describing what it is that we've searched for. So here we go. So this is the 54 different human morphologies that they have for CA1 pyramidal cells. Somewhere in here, there is, I can select all of these and I can download them together in one go. So if I want to say, give me all of these, I can do that. I can run whatever analyses I might be interested in on their links, their properties, whatever I want. How does the morphology of the human CA1 pyramidal cells what, you know, I don't know. What's the distribution of maximum path lengths from the sum? I don't know. Whatever you're interested in. How does this compare to that of the monkey? Well, you can literally download all of them, run it with Neuron, load in the morphologies, compare, and then do whatever analyses you want. And I will say that Giorgio Ascoli also has some tools for analyzing morphologies. Yes, sir.
Yes, I guess that's going to cover most part of my question. But if we want to specialize our model, like if we want to like individualize our model to the specific species, I believe we should go to the specific morphology, not using like an. And beside the morphology, what else we should consider?
Well, your question seems a little ill-defined. What exactly are you thinking of doing? You're thinking of making a model of a cell to study what phenomena?
Like say that I like to like to study human CA1 region in the hippocampus.
Okay. Well, there's not a whole lot of experimental characterization of cells from any part of human brain. Mostly because most people are kind of reluctant to give up chunks of brain, and it violates the Geneva Convention, I think, to take it away from somebody that doesn't want to give it up without their okay. So most of what we're going to find is going to be stuff that comes from people who have existing conditions that required resection. So it's going to be from people with epilepsy, where part of some overlying cortex had to be removed so that they get to a deeper cortical area, or maybe patients with neoplasms where overlying normal cortex had to be removed so they could get down to a thalamus or something that had something nasty growing into it. So you're not going to get very normal cells to begin with. There's not going to be a whole lot of electrophysiological characterization of these cells available. Certainly not if there's only 57 cells available for morphological studies. But you can try to find it in the literature. So you're going to have to make your best guess about what kind of biophysical properties to include from work done on other species. And there probably won't be a lot of work done on monkeys either because of restrictions on the kind of experiments that can be done on primates because of humanitarian concerns. So it's a challenging issue. I'm not saying it shouldn't be done, but it's going to be difficult. And your best bet would be to, if you're serious about working on this for any length of time, try to find somebody who actually has been engaged in experimentation on whatever species it is. Try to find, say, a neurosurgeon who has been involved in performing surgeries, or a neurologist who's interested in whatever condition you're thinking about, whether it's dementia or epilepsy or whatever, and try to let them know what your interests are, and see what information how they might be able to guide you toward finding available resources. I'm not saying things are not available. It's just that they will probably be rather scarce, and it may be it may take a lot of poking around to get results, okay, that are useful for you. When it comes to other kinds of animals, it's much easier to find empirical observations on rat and mouse than it is on humans.
Yeah, and so what I was trying to find but failing was, like I said, I know that there is some some people are trying to look at the differences between, and evolutionary differences between humans versus monkey or what have you. But even when I think I know one of the authors, I still couldn't find it. So
yeah, well, you would certainly start by looking at the papers from the labs that gathered this anatomical data. And that would be the first place I'd look. I would read those papers. So in Neuromorpho, the sources of all of these things and the papers where the data were published are mentioned. And I would not only just look that up, I would also look for who has cited their work and see if you can find experimentalists who are citing this paper, or maybe modelers who have been borrowing these data and using it in their models. I'd also think very seriously about what's been done at the European Human Brain Project, because I know that human brain pathologies are a strong motivating factor in that research project. And I'm sure that the people working on the European Human Brain Project have put a lot of thought into what kind of morphologies they're using, where they got them from, what kind of biophysical properties they're including in their models, where they got those from. Also, there is a huge amount of effort going now into genomic means of refining classifications of nerve cells and trying to reconcile genome and phenome. The problem with that work is, although it's very high quality work, and there's a lot of it going on, say, at the Allen Brain Institute and elsewhere, where they're very well funded and very smart people, they've been very clever about the machine learning algorithms that they've been using to try to identify unique markers for different cell classes. And as a result, they have found unique sets of markers for cell classes within a given species. But when you compare similar cell types across species, you find that the markers aren't conserved. In fact, almost none of the markers that are unique markers within any given gene, within any given species, have a whole lot to do with either the membrane biophysics of those cells or the synaptic transmitters or receptors that those cells have. So what we're dealing with there is markers that, yes, they may uniquely identify cell classes in particular genus and species, but they are only highly tangentially related to whatever connectivity and functionality those cells really serve in a brain. And I think that is a serious weakness. I mean, it's otherwise a very sophisticated, very well thought out work. I mean, it's a serious problem. If your unique markers don't actually, I mean, what is it that makes a hippocampal CA1 pyramidal cell useful to the animal is not that it has some unique marker that an anatomist can use to say, oh yeah, it's a hippocampal CA1 pyramidal cell. What makes it unique is its geometry, its topology, the afferents that project to it, the efferents that it projects out, the transmitters that it has, the ion channels that it has, the receptors that it has. And none of these things seem to be unique markers. So the unique markers are not capturing the essence of what it makes to be a CA1 pyramidal cell or any other kind of cell. Okay, so anyway, certainly somewhat frustrating. You know, it's like saying the most important feature about Robert McDougall is some immutable characteristic that has damn all to do with what functionality he serves in the world. Whether that be as a space occupier who holds down a chair, or a guy who cooks up these elaborate and interesting lectures. Okay, and it's trivial, unfortunately.
Yeah, go ahead.
Good luck to neuroscience.
Go ahead, Yusuf.
Yes, I don't want to make it a long discussion, but there are some like some literature from the human single unit studies showing that like in the place cells on the human are behaving totally different with the place cells on the rat, like in terms of the receptacles. So the question is that, is it like an individual cell differences between these two, or the connection, or both? So these are the things I like to know, that how I can include them in the model.
That's an extremely important, deep question. And I bet you dollars to donuts that that will take you years to discover. You'll find lots of interesting results and be able to publish a lot of papers. And in the end, you might be able to answer your question. But I'm glad you're asking it because it's a very perceptive and important question. It's not trivial at all.
All right, so just to, yeah, lots of things to think about. So just to go back to the morphologies directly and working with them in general within Neuron. So one of the things, remember we said it was great to create a cell class, use those 2 extra lines of code to make these templates that we can just stamp out. Instead of having one cell, I can have 10 or a dozen or 100 or 1,000 that all have the same morphology. So a while back, many, many slides ago, I had a representation of code that used the import 3D SWC loaded in this pyramidal cell. And then so I go ahead and I create my, my list of 10 cells. And then I bust out my shape plot. Doesn't matter if I do this, you know, I'm gonna get the same results whether I do this on code or graphically. And this is what I get. And I rotate this and I still only see this. So here's my question. So what happened? Clearly I can call the constructor 10 times. I should have made 10 cells.
Wait a minute. You actually rotated this cell?
Yeah, this cell I've rotated. This is a cell with 3D points, which makes a difference here.
Mm-hmm.
But this was the code that loaded this.
Okay.
I think there's a thing that would be different if you were doing a whole bunch of sections, but when we do 3D points, we rotated it and it's still the same. No matter how you look at it, you see 1, but there was supposed to be 10. So where are the other ones hiding? Well, I gave an X, a Y, and a Z and a diameter for every cell has exactly the same X, Y, Z's, and diameters because they all came from the 3D. They all came from the same representation that said this cell, this point on this cell is exactly here. So fine. So it
what
put
you mean
all.
is Neuron doesn't know anything about the Pauli exclusion principle.
As long as you're not talking about, there's actually a slight exception to that, which is with the chemical 3D intracellular simulations, and then having some things in the same space matters. But yeah, no, Neuron's perfectly happy with all 10 cells occupying the same volume because it doesn't see any connections there. So what you can do, and so here's a little bit of code that you can copy paste in your own things, but all this does is uses a 0.3D change. And what we do is we say we can modify our code ever so slightly, and we'll just put it somewhere else. You know, we'll put the ith cell at position I times 100, 0, 0. And then, so then there's a bit of an offset, right? The 0 cell starts at x coordinate 0, but the next cell starts at x coordinate 100. The next cell starts 200 microns out, and so forth and so on. So now when I look at the shape plot, or plot shape, depending on which term you prefer, they all appear separated by 100 microns. And incidentally, it suffices, as 2 other things to point out about this, it suffices to reposition the soma because everything is attached to the soma. And so if everything is in one place and I move the anchor point, then everything else is going to stay connected. It's all going to just slide along. The other thing that I want to point out is I'm also passing in an identifier in Neuron and also Arbor for that matter. These are GIDs, a global identifier. And basically giving ourselves numbers is extremely helpful in network models to be able to have a unique way of referring them so we don't have to talk about objects. We can just say like, the 5th cell, that one. And there's reasons that we want to do this. So but so here's some code you can copy paste for doing that. It'll put the cells, it'll translate the cells, it'll do the right thing. Here's my question for you though. Does it matter? Does it matter if the cells are, you know, what the shape is? Are there any let me put it this way. Are there any circumstances in which we do care about the shape and not just the path lengths and diameter, but like actually this branch is curved around? Does that matter?
When you talked about the RxD, you know, in your example at the beginning, I think that it would matter.
Oh yeah, a great deal. So if I'm talking about extracellular fusion, if I'm talking about 3D intracellular stuff, it's going to matter there. The other things, so even with the electrophysiology though, sometimes you're going to want to define rules. Sometimes you're going to want to define rules for when you're building up really sort of complicated morphologies. Maybe you make connections that are based on if an axon if you have a really elaborate axon representation, really elaborate dendrite representation. And one of the things that people do is they literally, sometimes some people grow these cells to based on statistical properties, basically reverse engineering. We have these 54 models of the CA1 human CA1 pyramidal cell. Can we identify, and we have this statistical distribution. So there are various algorithms that you can find in the literature. Giorgio Ascoli has one, Michele Maiori has one that can take all of this sort of data and generate synthetic morphologies. And so then maybe if you're dealing with these large networks, you want to base on what cells are near each other or even what axons pass near each other. And also,
electrical fields, local field potential generation, extracellular stimulation of cells with electrical fields. Shapes and locations are very important for those applications as well.
Absolutely. I should definitely expand this list. So there's lots of times when it might not matter, but there's a lot of times when it does. And it also, yeah, so it's not just making a pretty picture. It can have implications depending on the details of your model. So with that, the only thing that I have left is talking about the exercises. And again, I will post these slides momentarily after this lecture is done. So have access to them. There's 3 exercises again this week. But does anybody have any questions or comments on the material before we get to that? Okay, we've had a fair amount of questions during this, so okay with that. All right. So the first exercise is sort of to try and tie in all of the preliminary material together. So, we talked a bit about, so Hodgkin and Huxley didn't actually look at concentrations. Well, they didn't look at temperature either. But Neuron's implementation of the Hodgkin-Huxley model is temperature sensitive and uses the Q10 approach and blah, blah, blah. But so here's my question for you. So take that Hodgkin-Huxley cell. We've certainly implemented Hodgkin-Huxley enough times. Use whatever reasonable geometry you think, but do a parameter sweep on it and examine how, let's say, the action potential duration or any other electrophysiological properties that you're interested in depend on H.Celsius. And then while you're doing this, save it to a database using Pandas and use a separate script to actually plot the relationship. And so this is, again, it's extremely important to be able to assess parameter sensitivity because you often don't know the parameter values. Or sometimes, parameters do change over the course, between health and disease, and you want to see if you have a bifurcation going on somewhere in between. So parameter sweeps are extremely common, so this is inviting you to play around with the databases just because it gives you a system that scales for large problems. and also the EFL library for classifying things. So that's the first exercise. Second exercise is I've got 3 different morphologies. These are all CA1 pyramidal cells. I actually don't know what species, don't remember what species these are from, but you can look it all up in Neuromorpho, which is where all of these links will take you. So my suggestion for you is to take each one of these, download them, load them into Neuron, and check and see if you see any issues. Are they appropriate for use for simulation? Right? They're clearly good enough to be. They're clearly appropriate for whatever study they were. Every single one of these was was associated with a peer-reviewed publication. But is it appropriate for simulation? How do you feel about that? What do you like about each of these cells? What do you dislike about each of these cells? And my third exercise for you is, so here is one specific morphology, C91662. And I invite you to insert Hodgkin-Huxley channels in the soma and axon, passive channels everywhere. I guess I should, let's make it everywhere that's not the soma and axon, because the Hodgkin-Huxley channels already have the soma axon. Hodgkin-Huxley channels already have a passive channel included. But so go ahead and do this. Choose a reasonable discretization. I think we've seen code for using the d lambda rule, or just do any other approach that you think is reasonable. And plot the membrane potential, mid-action potential. And so this is the figure that I got. Obviously, your choices are going to make a difference as to what exactly this looks like. But yeah, so those are the 3 exercises that I would like to talk about. I encourage you to work on over the weekend, and we can talk about on Tuesday. Does anybody have any other questions on this? Hearing none, one final pitch. If anybody's going to the CNS conference, on Saturday, there's a Neuron and NetPine tutorial. NetPine is a level that sits on top of Neuron to sort of streamline the specification of network dynamics. So you'll see a sort of a different view on Neuron. I'll give a brief introduction to Neuron from a slightly different perspective, and then we'll talk about some of the chemical stuff a little bit more, and then move on to NetPine, which is a high-level specification of networks versus that might be of interest to you as well. So that's Saturday. It's one of the tutorials. You can see it on the CNS website. And I'll hang out for a little bit if there's any other questions. Otherwise, I hope everyone has a wonderful weekend.
Thank you.
Thank you.
Oh, I see you.